Good morning, everyone. Welcome to the Watchmaker Genomics talk on new chemistries for RT-qPCR and qPCR, focusing on the ability for fast extraction-free or crude lysis amplification for point-of-care testing. And I'm really happy to see some familiar faces out here and a lot of new faces that I don't know. Looking forward to meeting you after the presentation this morning. But I want to start by just kind of reminding everybody how we think about the needs of the point-of-care testing community when it comes to molecular diagnostics and amplification. And first of all, if we think about what everybody needs, faster turnaround, that's what we're trying to do. How quickly can we turn around a result? And along with that is the need to have inhibitor tolerance for your qPCR, RT-qPCR, because if you can be inhibitor tolerant, you can skip extraction or just do really crude lysis, everything like that. Get going faster, get to that result. Has to be easy to use. We would need to be minimal in the cold chain, so i.e. lyophilization. And then, of course, we want to be multiplexing detection as far as looking at multiple pathogens and everything that we're working on. But I want to back up briefly because if you know Watchmaker, you probably know us for our NGS applications. But what you may not realize is that we started out as an enzyme engineering company. And as part of that, we started this when we were thinking about NGS applications. But regardless of what we're doing, the capabilities and the platform that we have built in order to very rapidly take an idea and mutate that, create different variants, and screen that, and then really do deep characterization of those different variants, and then continue that flywheel as quickly as possible to generate new enzyme properties and new capabilities around speed, inhibitor tolerance, yield. Of course, we still want that specificity, sensitivity, and we're thermostability as well. And then what we're really trying to do is to do all of this in order to amplify samples in the background, very challenging matrices or challenging sample sequences. So where we started was with our StellarScript HT plus reverse transcriptase, which was really designed for some of our NGS applications. And that was designed for inhibitor tolerance and also very high heat tolerance, up to 65 C. We followed that up with release last year of our StellarTAC DNA polymerase, which is an engineered TAC polymerase, which was designed for very fast polymerization speed, again, going towards that, how quickly can we get to a result, but also high inhibitor tolerance, so building upon what we've already done with our HT plus RT. Talking about the StellarScript HT plus for just a minute, and I mentioned that heat tolerance. This is just an example of how we look at heat tolerance. And on the left-hand side, you can see a graph of what we're looking at, the aggregation and how quickly an RT aggregates and therefore starts to lose functionality as heat changes. So on the x-axis, you see lower temperatures up to higher temperatures. And then the two lines you see to the left are basically some of our more well-known RT enzymes that are out there and how when they start to aggregate compared to HT plus, which you can see doesn't start to aggregate until we get to about that 65 C. And then on the right-hand chart, what you see is a static temperature of 60 C over time. And again, how quickly some of our more well-known RTs start to aggregate compared to the length of time that it takes for StellarScript HT plus to do that. Just really hopefully kind of embedding that idea of just how stable this enzyme is as we get up to those temperatures. On the TAC side, we talked about fast amplification. So this is an engineered TAC DNA polymerase. And what we've compared it to, the first two bars are standard kind of wild type TACs that people work with. The third bar is another engineered TAC, and then the fourth bar is StellarTAC, in which you can see just how much faster we can actually incorporate nucleotides as we're amplifying, which again goes back to the speed at which you can amplify. And we think about these point of care type systems or these proprietary microfluidic systems that are just cycling extremely fast. You need a polymerase that can actually keep up with that and then amplify properly as you're working through it. So we had these two enzymes, but they were kind of standalone enzymes. And we said, how can we put this together in a real application for molecular diagnostics and the types of things that we're working with, and combine those characteristics of the enzymes to now be paired with optimized buffers designed for exactly these types of applications today. And what we looked at was two buffers, and I'll discuss those in a minute, with different properties, but we made sure that they're lyophilization compatible as well. So we're putting this together as a system with our point of care users in mind, so that they can design their assays, lyophilize those, and then still be able to optimize ahead of time, get really broad inhibitor tolerance, but then have separate components that allows you to dial in your assay. Because when you've got a master mix, there's really not a lot you can do to optimize your assay when it comes to the amount of polymerase, the amount of magnesium, all those type of things. So we've taken an approach to allow a tool set for our users to then design and optimize their assay for their needs. And as an example on the inhibitor tolerance side, so this is RT-qPCR, one-step RT-qPCR, using a 100-copy target as an example. And we've compared the Watchmaker solution to a large number of other well-known, vendor solutions that you can purchase out there. And what I want you to walk away with, and as you look at this chart, I realize you can't read the numbers, but look at the colors. So for this 100-copy target, anything in green is a CQ of less than 34. Anything in yellow is a CQ from 34 to 38. Anything in red is a CQ of 38, or if there's a dash, it means it failed, it didn't amplify at all, which is obviously a catastrophic failure and definitely what you do not want in this particular situation. And on the left-hand side, we've got a large number of inhibitors or biological samples. So we're actually spiking in whole blood or plasma or urine or universal transport media, things like that we know can inhibit your PCR. And we've also had some other inhibitors that are carryovers from extraction, like ethanol or guanidinium thiocyanate, showing that we can overcome those. And what I want you to walk away with as you look at this slide is the amount of green in the first two columns there. So that's Watchmaker buffer A and buffer B. And I mentioned we have two different buffers. And the reason for that is they have slightly different properties. And depending on your inhibitor, depending on how fast you want to go, the yield you're trying to look for, depending on the sequence you're working with, your primers and probes, you may find that one of those buffers works better for you than the other. And then you have that choice to which buffer you're going to work with going forward, and then to be able to dial in the amount of TAC polymerase, the amount of RT that you're working with. Similarly, on the qPCR side of things, again, 100 copy target. We're looking at the amount of green that's present there for the first two, compared to a broad number of other well-known solutions that are available and are regularly used in the molecular diagnostic space and point of care. So what I want you to walk away with is the high inhibitor tolerance that we're getting across the board with this system. Now, I mentioned speed as well, and this is a situation where we took an instrument called the OneSight X-Dive, which is a very, very fast qPCR instrument, and we wanted to push and see just how quickly we could get a result. I can tell you on this particular instrument, we were able to do 49 cycles in about 9 minutes, but even more impressive is that our 10-copy target, which is the lowest bar to the right there, the lowest line, we were actually able to detect 10 copies of a target in under 7 minutes, start to finish on that qPCR. So when we're talking about point of care and just how quickly you want to try to turn around a result, this is the type of thing you can achieve with our chemistry, provided you have an instrument that can actually cycle that fast. Now, all of this, as far as speed and inhibitor tolerance and everything, does you no good if you don't get a nice, range of, detection, your dynamic range. So here we're just showing 6 logs of dynamic detection on RT-qPCR, and this is a 3-target multiplex. We've separated these out so that you can see them, but anywhere from 10 up to 1 million copies, and you can see, clear, nice detection. And this was just literally taken straight out of the literature. We didn't optimize these primers and probes and everything else, so you can actually even get better results than this with a little bit of optimization with your particular target. Similarly, on the qPCR side, again, we took a 3-target multiplex straight out of the literature on human targets, and you can see just really beautiful, discrimination and distribution of your curves as you're working through on, 6 logs here. So going back to my original premise of what are the things that point of care assay developers need. So we talked about the faster turnaround time. We talked about the inhibitor tolerance, ease of use, and that cold chain situation, and then multiplex detection. And, here's kind of where we sit after doing all of that, in that we've got the enzymes that have that inhibitor tolerance capabilities that we discussed and that everybody needs, that's going to allow you to then, either skip extraction or go with a really crude lysis situation, which is, again, going to speed up your overall time to result, because you're going to just do something really quick, throw it in, do your amplification, and then as you've got these very fast cyclers, be it these cartridge-based microfluidics or anything else, our enzyme can keep up with as fast as you can possibly move those liquids around to actually do the thermocycling that you need. Now, when I talk about ease of use, I'm not talking about master mixes here. I'm speaking about the idea of the ease of use of assay development. Again, with a master mix, there's very little you can do with that. We are purposefully designed this system so that you can dial in the amount of polymerase, the amount of RT, the amount of magnesium into your chosen buffer, which will then allow you to create a master mix that's going to be perfectly optimized for the assay that you're developing to get the best possible results. On the cold chain side, every bit of this has been designed to be lyophilization compatible, so the buffers are lyocompatible. We have high concentration versions of these enzymes that are lyocompatible for you to develop this out. And then, of course, none of this would be useful if you didn't have the ability to do multiplexed qPCR or one-step RT-qPCR detection with the appropriate levels of sensitivity, specificity, and dynamic range that everybody comes to expect. And you're not going to give up any of that using a chemistry like this. I want to switch gears for the last few minutes of my talk and add in around another methodology that's getting rapidly used in the point of care and even more so in the point of need area, which is recombinase polymerase amplification, or RPA. And so we just actually in January also released a suite of RPA enzymes designed again for assay developers who are trying to use RPA for these point of need assays. And when I say point of need, I'm literally talking about being out in a field. I'm out in a third world country in a village. I'm out on a farm. I'm out doing aquaculture fish testing. It doesn't really matter. You're not in a lab. You're not in an office at any point. So this is what you're trying to do. And with RPA, you can actually get pretty fast results, anywhere from 10 to 20 minutes for the amplification, and then just generally run that on a lateral flow device. And it can actually be done at ambient temperature if you optimize the reagents properly. So, RPA will tell you that it's 37 to 42 C, but with proper optimization, we have lots of users who are doing this type of assay at ambient temperature, which makes it perfect for physically being out in the field, but still getting the sensitivity and specificity that a PCR type assay can do. And so it is also quite compatible with direct biological testing, so blood, saliva, things like that. I'll show you some data in a second. The field-based I talked about, everything, again, that we've developed is glycerol-free, designed for lyophilization compatibility, because if you're doing field-based testing, you absolutely have to have these reagents lyophilized in an assay that you've developed. And then, of course, we can actually do multiplex testing of either DNA or RNA targets, so you can do an RT-RPA reaction quite easily as well. So Why is RPA important? Most people are familiar with LAMP as an assay, but there are some really nice advantages using RPA over LAMP. First of all is primer design. It is much, much easier to design your primers for RPA. It's only 2 primers, 30 to 35 bases generally, versus 4 to 6 kind of crazy hairpin loop primers that are much more difficult to design. The incubation temp is much lower. Optimal, like I said, is 37 to 42, but you can do it at ambient. That is just something you can't do with LAMP without some sort of heat source to work for it. It's a little bit faster than LAMP, and the overall limit of detection is pretty similar between the two of them, but overall RPA is faster and simpler to run in the field. But as people developed RPA assays, one of the things that they ran into was a problem with consistency of the enzymes. And so this is one of the big innovations that Watchmaker brought to the table, was we are the first ones to figure out how to actually measure the activity level of the recombinase that's present in here, UVSX. And if you just buy it and use it by mass without understanding that the activity level has to be very consistent, then that can be really problematic when you create an assay and then you buy another lot and you buy it and you load it by mass, but at the same time the activity level isn't the same, and therefore your working assay isn't working like you thought it was before. So we have designed a way to give not only the same mass, but the same activity levels of these enzymes across the boards, which gives you much more confidence that you can build the same assay every time. And the other piece that we did is we designed, although everybody does this in a lyophilized form, we have designed these enzymes, to be freeze-thaw stable. So we've proven freeze-thaw stability up to 30 cycles of freeze-thaw, which means that you can work with these, do your development, not have to just thaw it and use everything in one shot and then waste it because it's not good after you refroze it. RPA as a general assay is, again, more of a qualitative. It is semi-quantitative, but most people use it as kind of a yes-no answer. But you can do like a real-time curve type of thing. They aren't quite as pretty as PCR. But in the end, what you're really looking for is, did you get a nice clean result band? And you can see on the right a tape station version of exactly what you see on the left. And you see anywhere from 1 million down to 100 copies in this particular case. And then from an inhibitor tolerance, the method overall is quite inhibitor tolerant. So here we're literally spiking in a variety of biological fluids and other things that could come through. So ethanol, and then you've got universal transport media, urea and urine, saliva, blood. So all the things that you might kind of, encounter work really well in RPA as a method. And then lastly, I want to touch very briefly, because I talked about the UVS activity, and that is something that activity level, not only being consistent, but just how much activity you have is extremely important. So we're comparing to a well-known other UVSX solution, and you can see on the left-hand side in the purple there, we have about 3 times the amount of UVS activity in comparison to another version of that enzyme that you can purchase. And then the other thing I would point out is that some people are varying between BSU and BST as the strand displacing polymerase that's used. If you're using BST, you physically cannot run at ambient temperature because the enzyme isn't really active. And if you're running at optimal RPA temperatures at 3742, which is what the right-hand graph is showing you here, the overall BST polymerization activity is extremely low in comparison to BSU. So if you're trying to do this in the field, you really have to have BSU, because that is the polymerase that can run at the optimal temperatures and would allow an ambient temperature run. So I will finish up here and just say, first of all, if anybody is interested, you can scan our barcode, and that'll take you to a web page that'll talk to you all about both of the things that I've spoken about here. But I will say our booth is directly behind you, back right over there, G37. So please feel free to stop by. But we'd love to talk to you more about the Stellar qPCR and our qPCR kits, which was the first half of my talk, as well as our RPA enzyme suite. And then lastly, for you, those of you who are assay developers, I want to let you know that Watchmaker is heavily involved in the OEM world as far as being able to develop these things for you, white label, do a lot of, partnership with you to get to your final result. So with that, I will stop. Happy to take some questions. I've got at least one of my colleagues around with a microphone. If anybody has a question, just raise your hand and we'll get to you. Thank you so much for coming. Yes.
Thank you for your talk.
Yeah.
I just have a really quick question about your extraction-free.
Yep.
What types of cells are we talking about? Is it also compatible with plants or animal tissues, or that one's requires?
So we have focused so far on more human samples. We are working to evaluate additional type of sample types. We haven't done that yet.
Okay, thank you.
Yep. Question at the back.
Thank you for your nice presentation.
Thank you.
Can you tell me something about? You already told that the polymerase is very fast, but what about the error rate? Does it produce?
The error rate is similar to a TAC. So yeah, it is an engineered TAC. The fidelity is same as any other TAC. All right, I think we have to cut it off because we're at time. Thank you for everybody coming. Appreciate it.